Welcome back to Mitochondrial Medicine 2021 as we continue with a clinical trials panel featuring discussions with Abliva, Baylor College of Medicine, Cyclerion Therapeutics, and Khondrion.
Hello, everyone, and welcome to panel 2 of our clinical trial updates for mitochondrial disease. In the session today, I'm joined by representatives from Abliva, Cyclerion, Khondrion, and Baylor University. Each representative will provide a brief update, and if time allows, at the end of the session, we'll also have a brief Q&A session. So let's get started. Our first speaker is Magnus Hansson. He's the Chief Medical Officer at Abliva AB, and please take it away.
So hi, everybody. As Phil mentioned, I'm Magnus Hansson. I'm Chief Medical Officer at Abliva. And at Abliva, our mission is to improve the lives of primary mitochondrial disease patients. We are a small biotech company based in Sweden, but we have an interesting portfolio specifically targeted to primary mitochondrial disease. So these are all innovative first-in-class therapies. None of them are approved, so they are in the drug development phase. And our most advanced project is KL1333, which just completed a number of phase 1 studies, including a phase 1b study in primary mitochondrial disease patients. So a little bit more about KL1333. So this compound has been designed to correct the underlying pathophysiology of primary mitochondrial disease, specifically the disbalance seen in NAD+ to NADH ratio. So the goal here is to improve energy production, both short-term but also long-term, by stimulating buildup of new mitochondria to compensate for the mitochondrial dysfunction. And the aim there is to change the trajectory of these diseases. So this is a highlight from the data from our phase 1a/b study. And this will also be presented as an e-poster by Dr. Rob Fitzheatley, which was the chief investigator of the patient part in this phase 1b study. So this phase 1 study included 64 healthy volunteers and 8 primary mitochondrial disease patients. And the most important part of it was to assess safety and pharmacokinetics. And KL1333 had a good safety profile. As exploratory measures, we included clinical outcome measures and biomarkers. And encouragingly, we saw signals of efficacy in this small, short trial in the clinical outcome measures explored. And also, interestingly, there was an association between exposure and efficacy. So looking forward, we are moving towards a phase 2/3 study later this year. And the patient voice has been really important in the design of this study. So a key endpoint will be fatigue, as this is a very common symptom across different primary mitochondrial diseases, and also ranked as being the most important for drug treatments to address in the voice of the patient report. So in preparation for the phase 2/3 study, we are doing a qualitative fatigue validation study. And this is actually performed in collaboration with UMDF, who helps us recruit patients. So this study will evaluate what is most important to address when it comes to mitochondrial disease fatigue, the symptoms and impacts on daily life. So we can address appropriate questions to be included in the phase 2/3 study. So the efficacy study will be commencing later this year, so look out for more specific information. So some specific details, it will be a 100+ patient study, and patients will be given either KL1333 or placebo for 12 months. Thank you.
All right, thank you, Magnus, for that update. And thank you for also reminding our audience that the virtual poster hall is also available to all meeting registrants, and that for many of the presentations today, there are more extended versions of the updates available. So please visit the virtual poster hall to find out more information. So thank you again, Magnus. So next up is Chris Winrow. He is the Vice President and Head of Translational Medicine at Cyclerion Therapeutics. And Chris is going to give us an update on their program. Chris.
Great. So thanks, Philip. Thank you to the organizers for the invitation to participate in the panel today. My name is Chris Winrow, and I'm at Cyclerion Therapeutics, which is a company that's focused on delivering new therapies for diseases affecting the central nervous system. I'm very glad to be here today representing a team of scientists, drug discoverers, and developers that have successfully brought forward the first molecule that's designed to target the soluble guanylate cyclase enzyme. This is a pathway in the brain that plays a key role in a number of critical processes, including mitochondrial function. And the new molecule that we've brought forward is called IW6463. So how does this molecule work? On the right-hand side of the slide, I'm showing a diagram. And IW6463 acts directly on the nitric oxide signaling pathway in the brain and the central nervous system. This is a pathway that's critical for the regulation of mitochondrial function and synthesis. And in patients with mitochondrial disease, dysregulation of the pathway contributes to impaired blood flow in the brain, oxidative stress, inflammation, metabolic crises, for example, stroke-like episodes, and cognitive impairment. And you can see from this diagram that citrulline and arginine are upstream within the pathway. It's important to recognize that citrulline and arginine have no direct action and are simply converted into nitric oxide. By contrast, IW6463 acts together with nitric oxide to stimulate soluble guanylate cyclase, or SGC, which is the key component of this pathway. And this actually amplifies the body's natural nitric oxide signaling. Stimulation by IW6463 with nitric oxide increases concentrations of a molecule downstream called cyclic guanosine monophosphate, or CGP. And this is at concentrations that are beyond what can be achieved following arginine or citrulline administration. CGMP is a molecule which is responsible for the beneficial effects of this pathway. At this point, IW6463 has been administered to over 100 healthy patients across 2 clinical studies in phase 1. We've conducted robust evaluation of safety, tolerability, and exposure, or pharmacokinetics, and found that 6463 is well tolerated, having good exposure within the plasma, but also within the brain. And we've also observed target engagement along with effects across a number of different measures of brain function. We've recently presented this information at a number of scientific conferences, most recently at the American Academy of Neurology General Meeting in April. And Chad Glasser is going to be at this meeting, the UMDF meeting, presenting a poster. We're currently conducting a clinical study in patients with MELAS. This is a study that's actively underway right now. We have up to 20 participants that will be enrolling to evaluate the safety, tolerability, and pharmacokinetics, the concentrations of 6463 in the brain, and also the effects on disease. This includes exploring potential effects on mitochondrial function, inflammation, blood flow, within the brain, neuroprotection, and cognition. To be considered for this trial, patients must be at least 18 years of age, have a genetic confirmation of mitochondrial disease, clinical features of MELAS, and elevated blood lactate concentrations during screening. There are other eligibility criteria that will also apply and be assessed at the clinical trial sites. This study is being led by key mitochondrial medicine centers of excellence. It includes the Children's Hospital of Philadelphia, Massachusetts General Hospital, Children's National Hospital in the Washington, D.C. area, Columbia University in New York, and Johns Hopkins University in Maryland. Scientists at Cyclerion also have a number of ongoing collaborations with mitochondrial disease experts, exploring the potential for additional opportunities for IW6463 beyond MELAS. Additional information on this study can be found at clinicaltrials.gov by searching for IW6463. So in closing, I want to thank the organizers of the UMDF and to convey the gratitude of the team at Cyclerion to all study participants and to all those who are working to find new therapies for patients and their families affected by mitochondrial disease. Thank you.
Thank you, Chris, for that update. Very much appreciated. Our third presenter in this panel discussion is Dr. Jan Smeitink. He is the founder and CEO of Khondrion, and Dr. Smeitink is going to provide an update on Khondrion's activities.
Okay, good day, everyone. My name is Jan Smeitink, and I'm the founding CEO of Khondrion. And before giving you an update on the development status of sondicormanol, our lead product, I would first like to congratulate the UMDF with its 25th anniversary. 25 years in which I've personally seen the progress made in primary mitochondrial disease R&D, and in parallel have seen the UMDF growing to where it stands now, a strong international-oriented organization promoting research and education for the diagnosis, treatment, and cure of mitochondrial disorders, and to provide support to affected individuals and families. And for the sake of patients, I do not wish the UMDF another 25 years to be needed, though sincerely hope that the efforts of all stakeholders involved made, the sooner the better, lead to breakthrough therapies, markedly changing the quality of life of all mitochondrial disease patients. So Khondrion is a privately owned biopharmaceutical company based in the Netherlands, a country formerly known for its tulips, wooden shoes, and windmills, but that view has luckily changed. With a focus on primary mitochondrial disease, Khondrion's initial target indications are mealy spectrum disorders caused by the maternally inherited 3243A2G mutation. Our lead product is sonlicromanol, also known as KH176. The compound has a unique triple mode of action, counteracting the three most important consequences of hampered oxidative phosphorylation. It's a redox modulator, a radical trapping agent, and has anti-inflammatory properties. The compound is supplied orally. Our clinical development program is steadily moving forward with a phase 1 study and a phase 2A study in mealy spectrum disorders already completed. A phase 1 study showing good safety, tolerability, and pharmacokinetics. A phase 2A proof of concept study showing improvements in alertness and mood. Important symptoms seriously affecting the quality of life of not only mealy spectrum disorders, but also other primary mitochondrial diseases. And based on the positive signals of the phase 2A study, we initiated a phase 2B study to reconfirm these positive findings. This study will be continued by an open label extension study to be initiated soon. As we really want to make contributions for all suffering from primary mitochondrial disease, we initiated also a phase 2 study in children with primary mitochondrial disease, of which the first kids have enrolled recently. The lessons learned over the past years show that cognition is frequently affected in patients with mealy spectrum disorders, that this causes a significant impact on their quality of life, and that in this group of patients with sonlicromanol and supported by extensive preclinical research, we observed a significant improvement in clinical and functional endpoints. The next 12 months, insightful clinical data will become available. These include the readout of the phase 2B in adults, initial data of the open label extension study, results of the phase 2A study in children, and additional data from a natural history study. All of these will bring us in a position to design and execute the best pivotal trials possible, hopefully leading to market authorization. And with that, I would like to thank you for your attention, and for more info, please visit www.khondrion.com. Thank you very much.
Well, thank you, Jan, very much for the update. Also, thank you for the kind words to UMDF, and without a doubt, we share your hope that there are breakthrough therapies developed very quickly for our mitochondrial disease patients. So thank you for your contributions on that front. Our final speaker in this panel discussion is Dr. Fernando Scalia at Baylor University. And Dr. Scaglia is going to provide an update on his citrulline trial being run currently. So turn it over to you, Fernando.
Thank you. I'm Fernando Scalia from the Department of Genetics at Baylor College of Medicine in Houston. I also want to congratulate UMDF for its 25 years. It's a wonderful organization, and we are very excited about this event. So today, I will focus on presenting this phase 1 citrulline clinical trial for adults with Miller syndrome. Miller syndrome stands for mitochondrial encephalomyopathy, lactic acidosis with stroke-like episodes. And typically, the affected patients have this A to G transition in MTTL1. And there has been previous work published in Japan by Koga that found that altered nitric oxide metabolism may lead to changes in the lining of blood vessels, decreased perfusion, and stroke-like episodes. And here in this brain imaging studies, we can see occipital and temporal areas being affected with metabolic strokes. And it's thought that this could be connected to mitochondrial proliferation that is found in the smooth muscle and endothelial cells of small blood vessels, such as these vessels that are surrounded by the intense blue on succinate dehydrogenase histochemistry. But moreover, we do know that arginine and citrulline act as nitric oxide precursors, arginine as a substrate of nitric oxide synthase, and then the byproduct, citrulline, in certain cellular types, colocalizes with two urea cycle enzymes, argininosuccinate synthetase and argininosuccinate lyase, and nitric oxide synthase for this de novo arginine synthesis, channeling this intracellular citrulline. And we do know, based on clinical use, that intravenous arginine supplementation leads to clinical improvement if given within a certain time of the onset of symptoms of metabolic strokes. Moreover, hypocitrullinemia has been reported in Miller syndrome. Therefore, a few years back, at Baylor College of Medicine and Texas Children's Hospital, we conducted stable isotope studies, which actually served as a pilot studies for this current clinical trial in adults and children with Millers. And we determined that both had impaired nitric oxide production rate when compared to control subjects, that both arginine and citrulline oral supplementation increased nitric oxide production, and that citrulline supplementation increased nitric oxide production to a higher degree than arginine supplementation. Therefore, we initially submitted this pilot as part of an AMDEC application, and it was funded by NIH as a pilot study, or now a phase 1 clinical trial. We had to go through FDA approval, and we obtained an IND for citrulline as well as NIH approval. And the primary aim of the study is really to establish the maximum tolerated dose of L-citrulline, oral L-citrulline, in 24 adult subjects with Millers harboring this common mutation at one of these following doses: 10 grams per day, 20 grams, 30 grams, or 40 grams per day. Subjects are enrolled one at a time, serially, and actually we have just recruited our first subject, and the first subject is currently receiving L-citrulline dosage of 10 grams per day. Our primary safety outcomes, or DLTs, those limiting toxicities, are associated with nitric oxide production, such as orthostatic hypotension, hypoglycemia, because arginine is synthesized from citrulline and is an insulin secretagogue, and headaches. The secondary aims is that we will determine cerebral blood flow and cerebrovascular reactivity using ASL MRI, and we are also monitoring guanidino compounds by metabolomic analysis, as guanidino compounds are derived from arginine. This trial is currently listed in clinicaltrials.gov with this NCT number, and we are pleased to say that finally, after the delays caused by COVID-19, we have recruited our first subject, and our current trial is active. So thank you very much. I really want to thank, and we want to thank UMDF Organizing Committee for giving us the chance to present an update on the trial. And if there are any questions, we have a clinical research coordinator, May Ali, that can be contacted. This is also being presented virtually in the poster session. Thank you very much.
Well, thank you, Dr. Scaglia, for that update, and again, the kind words. Really important for our audience to know that there's always more information to be found, both on our virtual platform, which all meeting registrants can watch on demand, but also at resources like clinicaltrials.gov. And so encourage our audience to use clinicaltrials.gov in particular to find out further details about these studies and potentially how they might be involved in them. So thank you collectively to the panelists for sticking to our guideline on time. That does leave us a few minutes for moderated Q&A. Magnus, I'd like to start with you. Your company is still at a relatively early stage with your drug development. And I wonder if you could just speak to the importance of engaging the patient community early in the process as opposed to later, perhaps whenever you're really doing a much larger recruitment.
Yeah, thanks, Phil. Yeah, well, I think for all of us on this side, it's really important to learn what really matters for the patient and also the specific nuances of those aspects. So often we are experts on how our compounds work and we know the general drug development requirements, but each disease area is unique. As you all know, primary mitochondrial disease are especially complex and heterogeneous. So, I mean, we have a deep interest in understanding how we can improve mitochondrial function and translate that to clinical relevant effects in patients, and selecting outcomes and how to target the development. That also has a bearing on which patients you include. So we need to select outcome measures that are feasible to assess in a clinical trial settings. There has to be enough patients for us to do the trial. And there are also multiple stakeholders. So, I mean, the regulators like the FDA, those that will pay for the therapy, our investors, but most importantly, patients. And to find something common there is, of course, a challenge. But if we start with the patients, that has the bearing on the rest of the stakeholders. Sometimes it takes a bit of education, but I believe that that's where we have to start, because then I think we'll come in with a good angle in the beginning.
Well, it certainly does speak to the importance of the patient voice. And, you know, I'm certainly happy that some of the resources UMDF has been a part of creating, like our externally led patient-focused drug development meeting, are a great starting point for anyone coming into the space to understanding the burden of the disease and what idealized treatments look like for mitochondrial disease patients. But there's always more, right, to be learned. And so applaud your organization for your activities with focus groups and surveys to try to better understand who it is you're serving with this. So thank you for those comments. Chris, I'm going to turn to you next. You know, for a company like Cyclerion, can you give our audience a little bit of insight as to how do you work backwards towards a target patient population, right? You know, you have a small molecule with certain biological activity associated with it. In your role, how do you work backwards? And in this case, how did you come to MELAS patients as a potential indication of interest for the compound?
Sure. It's an important question. I think identifying the right patients that would most likely benefit from a therapy is key, and doing that in a logical way and building it in a data-driven manner has really been key for us. I think it really starts with an understanding of a pathway and connecting that to the disease. And I think that can start through a number of ways, looking at bioinformatics approaches, looking at prior experience with therapies, and finding what might have had some promising results. And really using that to sort of drive the next stage, which is going into cells, whether they're patient cells or other cell-based assays, building the rationale there, continuing through the in vivo work that gets done. And I think coming out of that, you really are still left with a big question, and that's what's going to happen in people. And I think for us, we've taken a biomarker-driven approach, and really we understand that the pathway is connected. You heard from Dr. Scaglia, the importance of the nitric oxide pathway and experience there for MELAS in particular. So for us, a company that has decades of experience in nitric oxide SGC signaling, this really was an exciting opportunity. We brought forward the first molecule that gets into the brain directly and targets this pathway through SGC. And so for us, we sort of built that into the biomarker space. And so, moving into MELAS patients, We're doing exploratory studies right now to really understand what the potential benefit might be there, and bringing along the biomarkers that we've identified from those.
Yeah, so it's fair to say this is a case where everything aligned, right? You had a molecule familiar with the biological pathways it impacts, and then you can kind of start to work backwards, right, towards patient disease communities where it's known that dysfunction in that pathway, is pathogenic. So again, we wish you luck with your continued studies with your molecule.
I think, Phil, too, it's also, as you said, engaging with the foundations, UMDF. It was really helpful to us to understand what the needs of the patients and their families were. And so I think, through meetings and voice of the patient type activities, it's really helped us as well as we think forward.
Yeah, thank you for sharing that. So maybe to wrap up this session, we're very fortunate to have two experienced clinicians on the panel that are, of course, not here wearing their doctor jacket for the day, but more in their role as drug developers. But I'd like to turn to Dr. Smeitink and Scaglia, just for a brief kind of conversation about MELAS, right? So we've heard MELAS quite a bit in the presentations here in this panel. Dr. Smeitink, let me start with you. You're a clinician that has seen many patients, many MELAS patients. It's an attractive population for drug development because it's one of the more common adult forms of mitochondrial disease. At the same time, what might be some of the challenges associated with characterizing MELAS, because it's not homogeneous like most things in mitochondrial disease? Fair to say?
Yeah, that's absolutely fair to say, Phil. And it's an interesting and absolutely challenging question to answer indeed. So one of the challenging aspects I would like to mention is indeed the heterogeneity, which is present not only clinically and biochemically, but even in a homogenetic condition like MELAS, also genetically caused by the heteroplasmy phenomenon, the existence in variable ratios of both wild type and mutated mitochondrial DNA in the cells. And if we focus on that common mutation, which is classically described as MELAS, or mitochondrial encephalopathy, lactic acidosis, and stroke-like episodes, we now know, decades later, that this is clinically very heterogeneous. So we do not only have the classical MELAS syndrome, we have the maternally inherited diabetes, mellitus, and deafness, and various other oligo and monosymptomatic phenotypes. And that is also why we decided to talk about MELAS spectrum disorders, because it goes from classical MELAS to myopathy only. And that is certainly, if you talk about the design of clinical trials, that needs to be specifically addressed in the in- and exclusion criteria. The existence of this heterogeneity needs to be carefully addressed for proper patient stratification. And crucial to do so is the fact that important endpoints used for this and other indications are not validated yet for this particular group. And certainly, we also need to know more about the natural history of the disease for better stratification. And I know about the great activities the UMDF is exploring currently with respect to natural history. That should be a global initiative, in my view.
Yeah, I suspect we'll hear more about natural history as these panels progress. It is so important. Thank you for sharing some of the challenges of just characterizing something like MELAS. So Dr. Scaglia, we really only have a minute or so left, but you're part of what we would call an investigator-initiated trial, and I just wondered if briefly you could speak to our audience a little bit about the challenges of initiating a trial as an investigator at a university, as opposed to being part of a drug development company.
Thank you for that question. It's been quite challenging, and I think it has required persistent perseverance and grit to really continue with this.
You have all of those qualities.
Yeah. So initially, I think that we didn't know, and personally, I didn't realize that we were going to be required by NIH to get an IND from FDA. So that really set us on a path of like preparing even like a pre-IND meeting and a package submitted to FDA. Thankfully, I have to be very thankful. We have to be very thankful to Xiomara Rosales, because she worked at FDA before. She's obviously with NAMDEC, with the registry in Colombia, and she was an excellent advisor, and I think that really helped us prepare. But then there were like further questions. So that in and by itself took us 15 months to get an IND. And then obviously an NIH approval, which occurred, and working with the Data Monitoring Coordinating Center and getting a feasibility study at the Clinical Research Center at Baylor, and getting approval by the Clinical Research Center, an IRB approval. We were able to do all that. And when we were ready, we realized that the Data Monitoring Coordinating Center, the previous one, was going to be replaced by a new one. So we have to change all the forms, all the electronic case report forms, demographic forms, and we were doing that. And then last year we were ready to start, and suddenly COVID-19, we had to, so the clinical research unit closed. So it's been challenging. It's been challenging. I think that now it is very exciting to see this finally taking off after such a long time. Having our first patient coming. But I think that going back to that is also realizing that we needed third-party verification for citrulline. We needed to contact a company that was also willing to donate some of this compound to make sure that there was a stability and purity of the compound. And in addition, I think that I would also argue that there is, we may have been at the clinical site and without the support of a private sponsor, at some point we may have inadequate manpower. We may have even inadequate time for us, busy clinicians. Thankfully, there has been a full-time clinical research coordinator and a fellow. So between being on call, between clinics, trying to organize this, it has really required determination. So it's not easy. It's not easy.
Thanks for those insights. And we absolutely want you still taking care of our mitochondrial disease patients who find time to be in the clinic too. And really interesting topics. I thank all of the panelists for their presentations, for their feedback through the Q&A today. And we will be back shortly with panel 3 of our clinical trial updates. Thank you.
Coming up next, our clinical trial sessions continue with presentations from Astellas Pharma, Reneo Pharmaceuticals, Stealth Biotherapeutics, and Zogenix. Stay with us.